Okay, part 2. So let's go back to, let's see the, okay. Okay, carbohydrates and lipids. Okay, so, okay. Carbohydrates, organic molecules that contain carbon and hydrogen in 1:1 ratio. Okay, better way of saying that. CH2O. Carbohydrate, hydrated carbon. Okay, carbohydrate is the most the major energy source in the body. So, and this is even more true for microbiology. They function mostly on carbohydrates. We do too. We actually prefer carbohydrates. Okay, so it includes sugars and starch. The suffix OSE denotes sugar, so glucose. Okay, fructose, maltose, lactose, all that. Those are sugars. Okay. Okay. So the structure of carbohydrates. Sometimes they're written like this. Most of the time they're written like this. Okay, this is their usual form anyway. Okay. So a single unit is called a monosaccharide, and these are the monosaccharides. You take two monosaccharides, you put them together, you get a disaccharide. You start putting disaccharides together, you get polysaccharides. Okay? Okay. Starch, glycogen, glucose, or cellulose. Okay, all glucose is put together, but they're just put together in different patterns. Like some of them are flipped, some of them branch more. Okay. Starch is the plant storage unit of sugar. Okay. Glycogen is the animal storage version of sugar. Okay. Okay. Glycogen is sugar that's compacted together. So it doesn't draw in water and it doesn't form valence shells of water. So actually keeps the glucose all tightly packed. So it is denser. Okay. So it's easier to store glycogen than lots and lots of glucose. Okay. Okay. Cellulose is not digestible. So let me show you. This is glycogen. Okay. Okay. Okay, so this is dehydration synthesis and hydrolysis does not only apply to carbohydrates. It applies to pretty much everything in biology. Okay, the covalent bonds that hold monosaccharides together is called dehydration synthesis. I remove a hydrogen from one side, I remove an OH from the other side, and I just attach them together. Okay, so now I turn around and break that bond by adding hydrogen to one side and OH to the other side. Okay, so that is hydrolysis. Water, hydrolysis, break apart. Okay. Okay, so I remove water. Okay, to connect these two together. So dehydration, removal of water, synthesis put together. Okay. So this is dehydration synthesis. This is lysis, hydrolysis. Okay. I take that, I attach it to that to get water, and I end up taking this bond and attaching it to the other side of the O there. Okay. Dehydration synthesis. Oh, sorry, hydrolysis. All right, lipids. Lipids consist of non-polar hydrocarbon chains. Okay, since water is polar, fats are non-polar. Fats don't like water. Okay, water prefers other polar things or other charged things. Fats are neither charged nor polar. Therefore, the water tends to avoid fat. Okay. Okay, the most common type of fat you encounter is a triglyceride. Tri means 3. Okay, so triacylglycerols. Okay, so glycerol, the ol means it's alcohol. So the glycerol is a 3-carbon alcohol attached to that. So this is a triglyceride. So this is glycerol. I have 1 chain fatty acid, 2 chains of fatty acid, 3 chains of fatty acid. So triglyceride. Okay. All right. A fatty acid is a long polar hydrocarbon with a carbonyl group at one end. Okay. So. So this is a saturated fatty acid. So saturated means all the carbons are saturated by single bonds only, and there's no hydrogen that can possibly be added. A unsaturated fatty acid means I could potentially add hydrogens. So there are double bonds, and because they're double bonds, I can break the double bond and add hydrogen to them. Okay, that's what I'm sure you guys heard the term hydrogenated vegetable oil. Okay, hydrogenated vegetable oil is literally vegetable oil, which is unsaturated, and then they add hydrogen. So they break this bond and add 2 hydrogens there. They break that bond and add 2 hydrogens there. Okay. Okay. So when you draw this, these look the same, but they're not. In reality, they're not the same shape. They're more, when you have fats, they're usually, the bonds, the natural fats are usually cis double bonds. Okay, so they form kinks in the fat. Okay, these kinks in the fat allow them to keep spacing apart. Okay, it is hard to stack these things together. It's easy to stack these things together because they stack right on top of each other. Okay, so these things will be fats. These things will be oils. Okay, or the liquid, because liquids are further apart from one another. Solids are closer together. Okay, so the cis double bond allows them to be oils. The trans double bond doesn't. Okay, that's one of the reasons why trans fat is bad for you. The other reason why trans fat is bad for you is simple. We don't really know in our body how to break this double bond. We know how to break this double bond. We don't know how to break that one. Okay. Okay. Okay, keto bodies. Okay. Um, So this is a keto diet, right? Okay. So keto diet runs like this. You stop eating sugar, right? You cut sugars. That generates, so there's no sugar, so you burn fatty acids. Okay. Hydrolysis of the 3 formed fatty acids in the blood. These can be used for energy and converted to ketone bodies, where the keto diet comes from. Okay. Strict low carb diets and uncontrolled diabetes can result in elevated ketone levels. Okay, called ketosis. Okay. So ketone bodies. Okay. So this is a ketone body. It's also known as acetone, also known as paint thinner. So okay. Okay, phospholipids. Okay. Okay, phospholipids. Okay, phospholipids are lipids with a phosphate group. So let me show you what a phospholipid looks like. Okay, phospholipid. Okay, this is the lipid side. This is the phosphate side. The phosphate side is charged and therefore polar. The lipid side is non-polar, therefore hydrophobic. So you have a hydrophilic portion and a hydrophobic portion. And I'm sure you guys already know where this is going to lead. Okay, the hydrophobic portions will be away from water, and the hydrophilic portion will be stored water. You can put this together. Oops, I don't know what it is. You know, put it together. When you put them together, okay, you basically can make a cell membrane. Okay, so hydrophobic center, and the 2 sides are hydrophilic. Okay. They mention it here, but they'll show you a picture, which I was surprised. Okay, my cells. These phospholipids act as surfactants. Okay, they break down the surface tension of water. This actually allows your lungs from collapsing and allows you to breathe. Okay. Okay. Steroids are made from something called cholesterol. Okay, or cholesterol is a steroid. Okay, so here's cholesterol. Prostaglandins. Okay. So communication molecules. So prostaglandins. Again, they're really different from triglycerides, but they're still, you can see there's not a lot of OHs and stuff like that. Okay, so they're still hydrophobic. Okay. Proteins. Proteins are composed of an amino acid chain. Amino acid has a amino group, NH3, a carboxyl group, COOH, okay, and a functional group. Okay, the functional group is different from each amino acid and is usually referred to as the R group. Okay, there are 20 different amino acids, basically the 20 different functional groups. Okay. Okay. So the amino acids are charged to attract each other to form kinks and folds in the protein. Okay. Okay. The sequence of amino acids in a chain is ultimately determined by the DNA, and we'll talk more about that in another chapter. Okay, so here, okay, you have, this is your typical amino acid. You have a center carbon, you have a carboxyl group on one end, you have an amino group on the other end. Okay, this is usually negatively charged, this is usually positively charged. Okay, and then you have a functional group. Okay, so you can have non-polar functional groups, you can have polar functional groups, and you can have other things. Okay. Okay. When two amino acids are joined, the H is stripped from the amino end, and the OH is stripped from the carboxyl end. Okay, this is known as a peptide bond. Okay, so here you form a peptide bond. Peptide bonds are usually stronger than most other bonds. Okay, they have about a bond and a half strength of other bonds. Okay. 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 Proteins are important in their structure, and so we'll talk a little bit more about protein structure. Okay, so the protein structure is first determined by the sequence of amino acids. Which amino acids are in that sequence is the primary structure. Okay, the primary structure is literally the sequence of amino acids. Okay. The amino acids themselves will interact with each other, and they can form hydrogen bonds between them. And two of the most common ones are alpha helix and beta sheet or beta fold. Okay. So these are alpha helixes. You see how they're all connected here, like down the string? That connection is the primary structure. The secondary structure is the hydrogen bonding here. Okay, this is secondary structure. Okay, this is the alpha helix. This is the beta sheet. Okay. The combinations, okay, of alpha helixes and beta sheets put together the tertiary structure. Okay, the tertiary structure of the amino acid is formed by one polypeptide. Okay, sometimes that's it, and that's your protein. But other times your proteins are larger and they're made out of multiple polypeptides put together. And the putting together of those peptides is the quaternary structure. Okay. Okay. Since weak bonds hold the tertiary structure together, a protein is easily denatured by changes in pH and temperature. This is important. Okay. So remember, our blood has a very tight pH of 7.35 to 7.45. The reason why that's necessary is because if the pH is too far out of whack, okay, the proteins no longer work. And the same thing for temperature, which is why we have to hold to a very controlled temperature of 37 degrees. Okay. Okay, so the tertiary protein structure is made by weak bonds, weak ionic bonds here, some hydrogen bonding there, some van der Waals forces there. Van der Waals forces are they're the weakest type of bond. They're the hydrophobic interactions between carbon atoms. Okay. Okay, and then the quaternary structure. Okay. Okay. Sometimes proteins are combined with other molecules to become functional. Okay, so glycoprotein, like glyco is sugar, so glycoprotein, protein plus carbohydrate, lipoprotein, protein plus lipid. Okay. So you can see there. Okay. So lipoproteins, for example, when you measure like LDL or HDL, those are LDL literally stands for low density lipoproteins. Okay, structural, so protein function, structural function, collagen, okay, keratin, etc. Enzymes, they assist in chemical processes. So enzymes speed up a chemical reaction. Antibodies are part of the immune system. Carriers, we were talking about receptors, receive signals, usually hormonal signals and stuff. Okay, nucleic acids, okay? Nucleotides, or the building blocks for nucleic acids, they compose of a 5-carbon sugar, a phosphate group, and a nitrogenous base. Okay. So nitrogenous bases form into 2 categories, pyrimidines and purines. Okay. So a pyrimidine is a single carbon ring plus nitrogen, and there are 3 types: cytosine, thymine, uracil. The way I remember is pyrimidine pi, okay? Pi is a circle, so 1 single carbon ring, okay? And then what you do with pi's, you cut them. So C-U-T, okay? Okay. Purines are 2 carbon rings, okay? Adenine and guanine. The way to remember this, usually I just remember it's whatever the pyrimidine isn't, but pure, okay, and then Ag is silver, the scientific abbreviation for silver, so pure silver, okay. The pentose sugars that are attached, okay, pentose, 5, so pentose 5-carbon sugar, okay. Okay, DNA has A's, G's, C's, and T's. Okay, so I'm sure you guys learned that C's only bind with G's and T's only bind with A's. Okay, that is called Chargaff rules. And here you can see why, really. Okay, so C's and G's, okay? So first of all, notice the number of rings, okay? 1, 2, 3. 1, 2, 3. Okay, that makes them equal in size. So the 2 sides of the ladder are exactly the same. If I switch, like put these 2 together, they won't physically fit and you'll see a bulge in the ladder. If I put these 2 together, what you'll see is a indentation in the ladder, like it won't fit properly. Okay, that's one of the ways that we know which base goes with what. The other is the hydrogen bonds. Okay, so hydrogen bonds. Okay, so G and C's have 3 hydrogen bonds, A's and T's have 2 hydrogen bonds. Okay, okay, so there's a double helix. Okay, okay. RNA is closer to DNA, except it has ribose instead of deoxyribose. This is actually really important in biochemistry. That makes RNA much more unstable. It makes DNA much more stable, which is what DNA is, the genetic information, and not RNA. Okay. Single-stranded instead of double-stranded, it has uracil instead of thymine. Okay. Okay. So. All right. There are 3 types of RNA: mRNA, tRNA, and ribosomal RNA. Okay. Other RNA-related molecules serve important functions in the body. ATP is actually made from RNA. The adenosine is made from RNA. Okay. Cyclic AMP is also the same thing. It's made from RNA. Okay. NAD, FAD, these coenzymes are also made from RNA. Okay. Now, the last slide are some of the reasons why we understand there's an RNA world versus all the basic components of cellular machinery are actually RNA. Okay. All right, that ends this presentation.